Figuring out what kind of a crime may have been committed and who was involved is the most basic component to an investigation. If given sufficient supplies and samples, nucleic acids can be used to solve these questions. DNA methylation is an epigenetic concept where a methyl group is added to the 5' position of cytosine residues in CpG sites. Methylation-sensitive restriction enzymes and bisulfite DNA conversion are two common methods that can be used to analyze DNA methylation. Bodily fluids can provide crucial information and evidence about crimes that can be used for investigation. Fortunately, DNA methylation patterns can be used to identify bodily fluid samples. A study conducted by Fora and their colleagues looked into the methylation patterns in venous and menstrual blood, vaginal fluid, saliva, and semen to identify patterns that can be used as forensic material. At the end of their study, they concluded that the bodily fluids exhibited distinct empirical distributions of methylation percentages. Another study, conducted by Lee and their colleagues, looked into 5 tissue-specific differentially methylated regions in the genes DACT1, USP49, HOXA4, PFN3, and PRMT2. They found that for the DACT1 and USP49 genes, there was hypomethylation in the TDMRs for semen. Therefore, these two markers could be used to identify semen samples. Certain sites undergo hypermethylation or hypomethylation as an individual ages. DNA from many different individual sample types can be used to determine age, including blood, semen, and saliva. A study conducted by Bauckland and their colleagues identified 88 sites in or near 80 genes where the degree of cytosine methylation had a significant correlation with age in the saliva of their sample population. They also validated methylation pattern changes with age at 3 gene promoter region sites with another sample population. The genes EDAR-ADD, TOM1L1, and NPTX2 showed methylation patterns that were linear over time. These studies proved that there are age-related patterns in regards to DNA methylation that can be recognized and used to help in identifying individuals in forensic investigations. Using nucleic acid applications, specifically DNA methylation patterns, is growing in the field of forensic science, 2 of the most prominent applications being age determination and bodily fluid and tissue determination. As more research has been conducted and new technologies have been developed, nucleic acid analyses have proven to have suitable applications in the forensics scene.
Created using Powtoon.